right, so in this case, I'll start then. My name is George Zachos, and I will talk to you about two novel signaling pathways that we identified in my lab in the Department of Biology in Greece, in the University of Crete. So these pathways prevent chromatin breakage in cytokinesis in human cells. So during mitotic cell division, the cell must accurately divide its genetic material, its chromosomes, into two daughter cells. And the final stage of the cytokinesis is called abscission. It's the stage where the narrow intercellular canal that connects the two daughter cells is cut to release the daughter cells. So in late cytokinesis in cells with normally segregating chromatin, several proteins of the X-cord family, including CHAM4C that we'll see later, and also the ATPase VPS4, are recruited to the mid-body, and they mediate abscission to release the two daughter cells. However, sometimes chromosomes become tangled. When this happens, a chromatin bridge can arise. Chromatin bridges are strings of chromatin that connect the two daughter nuclei, and when they persist in cytokinesis, they can cause two things. First, after abscission, a chromatin bridge can break, so then we will have DNA damage, or a chromatin bridge in cytokinesis can lead to furrow regression and formation of tetraploid cells, and both these outcomes can lead to chromosomal instability, and which is associated with cancer predisposition. So the cell has a regulatory mechanism which is called the abscission checkpoint, which stops abscission in the presence of chromatin bridges to maintain chromosomal stability. The mitotic kinase Aurora B mediates the abscission checkpoint at the mid-body. However, the molecular mechanisms that are involved are incompletely understood. So a few things about Aurora B. It's a conserved serine threonine kinase. It's a part of the CPC protein complex that also has three other proteins, survivin, borealin, and insulin. Aurora B is phosphorylated at serine 331 from other kinases, and this phosphorylation is essential for its maximal catalytic activity. And during cytokinesis, the entire CPC complex localizes to the mid-body and regulates abscission timing. Some other kinases that I will talk to you about are the clock kinases, CDC-like kinases. So clocks 1 to 4 in human are evolutionary conserved kinases. They localize in the nucleus where they regulate alternative splicing, but their role and their biological targets are really incompletely understood. So a few years ago, we developed an interest in studying cells with chromatin bridges. This is a confocal microscopy image of a control human carcinoma cell. And you can see in blue the DNA, and these are the two daughter nuclei connected by a chromatin bridge, that you see here. In red, we see SNP, a protein that also marks the mid-body. And the green channel is clock2. So surprisingly, we noticed that clock2 localizes to the mid-body in cytokinesis with chromatin bridges in control cells. And similar images were obtained for clock1 and clock4. So clock1, clock2, and clock4 go to the mid-body in cytokinesis with chromatin bridges. And also importantly, if you deplete clock1 or clock4 by siRNA, you can see a reduced localization of clock2 to the mid-body compared with the control cells. So I should say that the insets are magnified mid-bodies. So clock1, clock2, and clock4 localize to the mid-body as a complex in cytokinesis with chromatin bridges, and this raises the possibility that they have a role in maintaining the chromatin bridge integrity. So to investigate this, we measured the percentage of broken DNA bridges in cells depleted for clock 1, clock 2, or clock 4, or cells treated with a clock inhibitor, TG003, you can see that clock deficient cells have a high frequency of broken chromatin bridges compared with control cells. And these are examples of broken chromatin bridges in clock deficient cells. The dotted arrow indicates broken bridges. And this is a control cell with an intact chromatin bridge. So clocks 1, 2, and 4 prevent chromatin bridge breakage and cytokinesis in late cytokinesis. Also importantly, when we transfected cells with a dominant negative VPS4 mutant fused to GFP that inhibits abscission, we noticed that cells transfected with this mutant, which is a black bar here, so cells that cannot go to the cannot complete abscission, exhibit reduced chromatin bridges, broken chromatin bridges, compared with the GFP-only controls in the absence of clocks, which shows that clocks prevent chromatin breakage by acting in the abscission checkpoint. Aurora B, as I already told you, is a main regulator of the abscission checkpoint in human cells. And phosphorylation of Aurora B at serine 331 is a marker of Aurora B catalytic activity. So we investigated a localization of phosphorylated Aurora B serine 331 in cells with chromatin bridges of the mid-body. Green is phosphorylated active Aurora B, red is the mid-body, you can see it here, and the blue is the DNA. So in control cells, we see phosphorylated active Aurora B at the mid-body. Clock deficient cells, however, exhibit reduced localization of phosphorylated Aurora B at the mid-body compared with the controls. And this is also quantified here. This graph quantifies the intensity of active phosphorylated Aurora B at several mid-bodies in clock deficient cells compared with the controls. So clocks are required for Aurora B serine 331 phosphorylation and Aurora B activation at the mid-body in cytokinesis with chromatin bridges. Active Aurora B phosphorylates CHAM4C at serine 210 to prevent abscission. Therefore, we investigated also CHAM4C serine 210 phosphorylation. Green here is phosphorylated CHAM4C, red is the mid-body, and blue is the DNA. You can see that in control cells with chromatin bridges, phosphorylated CHAM4C localized to the mid-body. Clock deficient cells, however, exhibit a reduced phosphorylation of CHAM4C at the mid-body compared with the controls. So clock catalytic activity is required for CHAM4C serine 210 phosphorylation at the mid-body. Also, when cells were transfected with a phosphomimetic CHAM4C GFP, in which serine 210 is mutated to aspartic acid, so transfection of this phosphomimetic CHAM4C GFP rescues broken bridges in the absence of clock activity, which is the black bar here, compared with the control cells, whereas transfection of a non-phosphorylatable serine 210 to alanine CHAM4C mutant, that you can see here, induces chromatin breakage in control or clock deficient cells, similarly. So these results show that phosphorylation of CHAM4C in serine 210 prevents chromatin breakage in clock deficient cells. So from these and other results, we made this model. We proposed that clocks 1, clock 2, and clock 4 localize to the mid-body in cytokinesis with chromatin bridges. They phosphorylate Aurora B at serine 331 at the mid-body to activate Aurora B, and active Aurora B phosphorylates CHAM4C in serine 210 to prevent abscission and prevent chromatin breakage in cytokinesis with chromatin bridges. And this model we published a few years ago in Nature Communications. So another thing that happens in control cells with chromatin bridges, apart from activating the abscission checkpoint, is also that they form these actin patches, these accumulations of polymerized actin at the base of the intercellular canal that contains the chromatin bridge. So these actin patches, we think that they provide mechanical stabilization on the chromatin bridges, perhaps by counteracting the opposing forces that are exerted on the bridge by the daughter nuclei. However, how exactly the actin patches are formed has not been previously shown. So, CHEK1 and SRC kinases, we found that they play an important role in making these actin patches. CHEK1 is a conserved second threonine kinase that is very well established as a regulator of the DNA damage and DNA replication responses in human cells. SRC kinase is a non-receptor tyrosine kinase that is involved in actin nucleation and actin dynamics during several processes such as cell migration and cell spreading. And we visualized actin patches in control cells, as you can see here, microfocal microscopy. So green is actin, and blue is the DNA. And you can see a control cell with a DNA bridge here. And these are the actin patches, these accumulations here that are shown by arrows and are also magnified in this instance. The numbers here indicate the relative intensity of the actin at the base of the canal. So control cells form actin patches, as you can see, whereas remarkably, cells without SRC, cells transfected with SRC siRNA or CHEK1 deficient cells, do not form actin patches. You can see that actin patches are much lower here in SRC deficient cells or in CHEK1 deficient cells compared with the controls. And this is quantified here, where we quantify the intensity of actin patches in SRC deficient, CHEK1 deficient cells or cells treated with a SRC inhibitor called PP2. And the reduced actin patch formation in SRC and CHEK1 deficient cells also correlates with chromatin bridge breakage in these cells compared with the controls. You can see broken bridges here. So these results show that CHEK1 and SRC are required for actin patch formation in cytokinesis with chromatin bridges. And we wanted to investigate how they do that. We visualized SRC in control cells with chromatin bridges. So blue is the DNA, red is actin. You can see the actin patches here and here magnified. And the green is SRC. You can see that SRC localizes to actin patches in control cells, which indicates a direct role for SRC in actin patch formation, whereas CHEK1 does not localize to the actin patches, which suggests an indirect role for CHEK1 in actin patch formation, perhaps by regulating SRC. So since CHEK1 is a kinase, we investigated whether CHEK1 whether it phosphorylates SRC. To investigate this, we did in vitro kinase experiments with purified CHEK1 and SRC proteins and radioactive gamma ATP. And we noticed that CHEK1 phosphorylates SRC in vitro. And when we analyzed by mass spec the phosphorylated SRC, we identified a novel CHEK1 phosphorylation site on SRC, serine 51. And you can see that mutating serine 51 to alanine diminishes SRC phosphorylation by CHEK1 in vitro. So CHEK1 phosphorylates human SRC on serine 51 in vitro, and this site is conserved in higher eukaryotic cells. To investigate the significance of this phosphorylation, we did immunoprecipitation kinase experiments using cells transfected with GFP SRC constructs and using phosphorylation of FAC tyrosine 925 as a substrate. And you can see that non-phosphorylatable serine 51 to alanine SRC exhibits a reduced catalytic activity against our substrate compared with the wild-type SRC or the phosphorylated SRC, which shows that serine 51 phosphorylation by CHEK1 is required for optimal SARC catalytic activity. We raised a phosphor-specific antibody against phosphorylated SARC serine 51 to investigate whether this phosphorylation also happens in cells. So as you can see here, these are control cells. Green is phosphorylated SARC serine 51, red is actin, blue is the DNA. You can see that phosphorylated SARC serine 51, it's better shown here in the insets, localizes to the active patches, okay, at the base of the bridge, in control cells, whereas CHEK1 depleted cells do not show SARC serine 51 phosphorylation and also do not form active patches. So CHEK1 is required for SARC serine 51 phosphorylation in cells. And finally, when we transfect cells transfected with GFP SARC serine 51 to aspartic acid, which is the phosphomimetic mutation, you can see that cells transfected with phosphomimetic SARC exhibit actin patches in the absence of CHEK1, which is the black bars here, compared with the controls. And also, the phosphomimetic mutation rescues broken bridges in CHEK1 depleted cells, the black bars, compared with the controls, which are the white bars. So phosphorylated SARC serine 51 promotes actin patch formation and prevents chromatin bridge breakage in late cytokinesis. So these and many other results that I don't have time to show you today led us to propose the following model. CHEK1 phosphorylates SARC at serine 51, and this phosphorylation promotes complete SARC catalytic activity, and active SARC participates in several cellular functions, and also in the presence of chromatin bridges, active SARC, phosphorylated SARC, goes to at the base of the intercellular canal, promotes formation of the actin patches, and promotes stable chromatin bridges in cytokinesis. And this model we published in Journal of Cell Biology a couple of years ago. So to summarize, I gave you a taste today of our findings regarding two signaling pathways that prevent chromatin bridge breakage in cytokinesis. One is the CLOCK1, CLOCK2, CLOCK4 Aurora B chamfer pathway that prevents abscission, and the other is the CHEK1 SARC pathway that promotes actin patch formation. Both these pathways lead to prevent chromatin breakage in cytokinesis and ensure chromosomal stability. So I'll stop here and I'll thank people in my group that participated in this work. I also thank our collaborators for the mass spec analysis, and also Fondation Santé, Worldwide Cancer Research, the Bodosaki Foundation, and the Greek State Foundation for funding our work. Thank you for